In order to prepare for the BIOPAC ECG Lab, you must first complete the ECG Pre-Lab outlined in the Bio 112 Must Know Lab Manual prior to the lab. The first step outlined in the Pre-Lab is to download the latest version of the free BIOPAC Student Lab application from the BIOPAC website. Step-by-step downloading instructions are listed on ECG page 2 in your Must Know book. If you have any trouble following these steps or prefer a video demonstration with troubleshooting tips, watch the How to Download BIOPAC video tutorial showcasing these steps on the MC Anatomy Physiology YouTube channel. A link to this site can also be found in Canvas under the AMP2 Lab Resources section. If you do not have a computer, however, or are unable to download the application to your computer, the South Hall Upstairs Computer Lab has the free software already installed and can be used to complete the remainder of the pre-lab. If you haven't already downloaded the BSL application, you can pause this video here to complete the downloading steps before continuing. Once the BIOPAC Student Lab application has been downloaded, you can then complete the remainder of the ECG Pre-Lab. This entails opening the BSL application and following the instructions on ECG page 3 in your Must Know book. A window will then open with a PDF of the ECG lesson introduction. As outlined in your Must Know book, you must read through this, as well as the ECG lesson prologue on page 4, and watch the remainder of this video in order to answer the ECG Pre-Lab questions correctly on pages 5 and 6 in your Must Know book. Once you've completed the Pre-Lab in full, you are then ready to conduct the ECG Lab. On the day of the ECG Lab, you will be placed in groups of 3 to 4 students. Each group will be provided the following: a BIOPAC MP36 unit, a recording laptop with power cable, a labeled BIOPAC equipment satchel, a yoga mat, and a set of ECG Lab Procedures printed and bound. Please note that these printed instructions must remain in the lab for other sections to use. However, a PDF version of these printed instructions can also be found in Canvas under the AMP2 Lab Resources section. Pages 1 through 12 of these instructions detail how to record an ECG for each test subject in your group, which is what you will be doing in the first half of the lab. Once all ECG recordings for your group are complete, pages 13 through 27 then detail how to open your file on a separate computer to complete data analysis for your ECG recordings and answer the ECG post-lab questions in your Must Know book. Keep in mind that you will only be responsible for analyzing your own ECG recording. If you cannot record an ECG for medical or personal reasons, speak with your professor before the lab. Position the BIOPAC MP36 unit and BIOPAC recording laptop at one end of the lab table near an outlet. Plug in the laptop power cable and turn the laptop on, hitting enter for the password when prompted. Open the BIOPAC satchel and remove the BIOPAC power cable from the back compartment. This cable is comprised of 2 parts. Ensure the cable is connected firmly at its adapter. Then plug one end of the MP36 power cable to the outlet at the end of the lab table and the other end to the DC input on the back of the unit under the DC input label. Ensure that the unit power switch is down and the unit remains turned off while you connect the remaining cables. You can confirm this by checking to make sure the green power light is not lit on the front panel under the power label. Now remove the wound USB cable from the USB compartment of the BIOPAC satchel. Note how this cable has 2 different connection ends. Plug in the flat USB end to an empty port on the laptop. Plug in the male type B connection end to the back of the MP36 unit under the USB label. Be sure to match the shape of the cable connection properly here. If this cable end is forced in and plugged in upside down, the laptop will not be able to connect with the BIOPAC unit. Now remove the wound electrode lead set cable from its labeled compartment in the BIOPAC satchel. Plug in the pronged connection end with the label facing up under the channel 1 or CH1 port on the front of the unit. Once plugged in, turn the side screws all the way down to secure the cable in place. Nothing should be plugged into the electrode check port or channel ports 2 through 4 on the front of the unit. With all cables properly plugged in, now turn the power switch on the back of the BIOPAC unit on. The green power light should then light up on the front of the unit under the power label. If the power light does not come on, check the power cable connections at the MP36 unit, the outlet, and adapter. this doesn't correct the issue, switch ports on the outlet or ensure that the test button hasn't been accidentally pressed. Lastly, roll out a yoga mat near the end of the lab table, keeping it near the BIOPAC unit but giving enough space for your group to stand in front of the recording laptop without stepping on the mat. Following the instructions in the printed ECG Lab Procedures manual provided, prepare a test subject for recording. Have the subject lie down in a supine position on the yoga mat. If their shoes cover their ankles, ask that they remove them. If the subject has any jewelry on their right wrist or either ankle, also ask that they remove them so that they don't interfere with the recording. Remove an electrode from the plastic strip and check the underside foam. If the foam covering the electrode appears dry, press the nozzle of the gel tube into the foam of the electrode and squeeze a small dab of gel into the foam. Do not squeeze a mound of gel over the top, as this will spread to the white sticky ring and the electrode will not stick properly to the subject's skin. Place one electrode on the medial surface of both the left and right ankle, 1 inch above the medial malleolus. Then, place one electrode on the anterior surface of the right forearm about 2 to 3 inches above the wrist. The electrode lead set cable attached to channel 1 has 3 color-coded clip ends. Attach the electrode cable ends to the electrodes on the subject, following the color code shown in the lab instructions. Ensure the cables have been properly attached and the electrodes are not coming off before proceeding. Open the BIOPAC Student Lab application on the desktop of the recording laptop. If the BIOPAC registration window opens, just close it out and proceed to the application. Select Lesson L05 Electrocardiography ECG 1 from the menu list and click OK. Then type in the subject's first name as the file name in the window that opens and click OK. To protect the subject's privacy, if they do not want their name associated with their ECG recording, you may type an alternate name of their choosing. You are now ready to begin calibration. Calibration requires an 8-second reading of the subject's ECG to ensure proper electrode connection. This is not a recording, just a way to confirm everything is attached and running properly. Every subject in your group will be required to do this before the first recording. Read through all the calibration steps in the Lab Procedures manual before beginning. Ask that the subject remain relaxed with their eyes closed and that they do not speak or move while calibration is in progress. Click Calibrate at the top of the screen and wait for it to stop automatically after 8 seconds. You should see a clear ECG pattern repeat on the screen during this time. If the ECG pattern is unclear, click Redo Calibration and review the troubleshooting checklist in the lab instructions before repeating steps 1 through 5 and redoing calibration. If the ECG pattern is clear, however, and you can make out the P waves, QRS complexes, and T waves, click Continue. You are now ready to record the first of 4 experiments in the ECG lab, the supine condition. Read all the supine recording steps in the printed Lab Procedures manual before beginning. Similar to calibration, ask that the subject remain relaxed with their eyes closed and that they do not speak or move during the recording. When ready, click Record and keep an eye on the running seconds bar on the bottom of the screen. You will want to record supine for 20 seconds. If the ECG looks a bit flat, you can improve the display of the waveforms during the recording by selecting Display from the top menu and then Auto Scale Waveforms from the menu list. When 20 seconds has been recorded, you can then click Suspend. After hitting Suspend, verify that the recording resembles the example data in the lab instructions. You should see a clear ECG waveform pattern in the top recording strip and a heart rate line with little variation in the bottom recording strip. If the ECG is unclear, click Redo and review the supine recording instructions before repeating supine steps 1 through 5. If the ECG pattern is clear, however, click Continue to proceed to the next experiment. Be sure to click Continue and not Done at this time. As is warned in the lab instructions, if you click Done before all 4 experiments have been recorded for your subject, you will have to exit the program and restart the ECG lesson from the calibration stage. After clicking Continue, you will notice that the title of the previous recording remains at the top of the screen while the bottom half of the screen showcases the name of the next experiment you are about to perform, which is the seated condition. Review all the recording steps so that the subject knows what to expect before beginning the seated recording. If you haven't already, place a chair near the test subject and then ask that they stand up onto their feet and quickly and carefully sit on the chair with their hands on their lap and feet on the floor or the lower bar of the stool. Assisting group members should ensure that all cable connections remain in place while the subject is moving. As soon as the subject is seated and still, immediately click record. Keep an eye on the seconds bar below the recording strip. This will begin where the previous supine recording was paused. After 20 seconds of the seated condition is recorded, click suspend to pause the recording. In this example, the recording begins at 24 seconds and is paused at the 44-second time mark. If an error occurred during the recording and the ECG waveform is unclear, click redo and repeat the seated condition steps, beginning with the subject back in the supine position down on the yoga mat. If the recording resembles the example data, however, and there's a clear ECG waveform pattern and a slight variation in the heart rate line, click continue to proceed to the third experiment, the deep breathing condition. Read through the deep breathing recording steps in the printed ECG Lab Procedures manual carefully with the subject, and practice the breathing count outlined in the instructions before starting. The subject must inhale and exhale slowly and fully at an equal and comfortable pace. This pace will be determined during the practice session advised in the instructions. Here's an example.
Inhale, 1, 2, 3, 4, 5. Exhale, 1, 2, 3, 4, 5. Inhale, 1, 2, 3, 4, 5. Exhale.
When ready, click record and have an assistant group member begin the breathing count in the instructions aloud, so that both the subject and the recorder can hear. When the assistant tells the subject to inhale, the recorder must press the F4 key so as to insert a start of inhale arrowhead marker above the recording as it's running. When the assistant then tells the subject to exhale, the recorder must then press the F5 key to insert a start of exhale arrowhead marker above the recording. This count will continue for 3 breathing cycles. Continue to alternate between pressing the F4 key at the beginning of every inhale and the F5 key at the beginning of every exhale as the assistant tells the subject what to do. Once 3 breathing cycles have been recorded, regardless of the time on the running seconds bar, click suspend to pause the recording. You should have 3 inhale arrowheads and 3 exhale arrowheads visible over the ECG recording waveforms. If they are missing, or any error occurred during the recording and the ECG waveform isn't clear, click redo and repeat all the deep breathing condition steps. It is normal, however, to see variation in the heart rate recording strip. If the recording resembles the example data and arrowhead markers were properly inserted, click continue to proceed to the next experiment. The fourth and last ECG experiment is the after exercise condition. As always, read through all the printed instructions before beginning. Unclip the electrode lead set cable ends so that the subject can move freely. The electrode stuck to the skin, however, should remain in place. Ask the subject to select an exercise to perform for 1 minute. A list of suggested exercises is in the instructions. After 1 minute of exercise is complete, have the subject sit back down in a chair while assistant group members reattach the electrode cables according to the color code in the lab instructions as quickly as possible. As soon as the cables are reattached and the subject is seated and still, immediately click record. The after exercise condition must be recorded for 60 seconds, so take note of the start time on the running seconds bar when it begins. In this example, the recording is started at 78 seconds, so it should be suspended at 138 seconds. To improve the view of the ECG waveforms and heart rate line, you can once again select Display from the top menu and then Autoscale Waveforms while the recording is in progress. During this time, ask the subject not to speak or move, but sit calmly and return to normal breathing. Once 60 seconds has been recorded, click Suspend. If an error occurred during the recording, click Redo and let your subject rest for several minutes before repeating the after exercise recording steps. Keep in mind that with an increase in heart rate, the ECG may look erratic or compressed as it recovers. If an ECG pattern is distinguishable in most of the recording, as is the case in this example, click Continue or Done to keep the recording and move on to saving. If you click Continue, you can then click Done on the next screen. Note that you may only click Done now, being that this is the last recording for the subject. Follow the saving instructions in the printed ECG Lab Procedures manual provided carefully. This must be done after each test subject in your group has recorded to ensure data is not lost. When you click Done, a window will appear asking if you are sure you are done recording for this test subject. Click Yes. A BIOPAC Student Lab window will then appear asking what you would like to do. Select Copy to another location from the menu and click OK. Please note that if you make any other selection at this time, the entire ECG recording could be lost. After selecting Copy to another location, designate a location to save the file to in the window that opens. If you don't have a USB drive, select Desktop on the left of the window and then click Select Folder. Do not type anything in the folder field. The file will be saved under the subject name that was typed in at the very beginning of the recording. If you have a flash drive, however, you can plug it in and select it on the left of the window to view its contents before clicking Select Folder. Again, do not type anything in the folder field. To visually confirm that the file saved successfully, you can then click the File Explorer icon on the bottom of the screen as shown and open either the flash drive or desktop. If you see the file name there, then it has saved to the location you selected. Do not attempt to open the file from here, however. It can only be opened via the BSL analysis application that you downloaded as part of your pre-lab. This step is just to make sure the file saved before you record the next subject. Once the file is saved, the BIOPAC Student Lab will again appear. Now select Record from another subject from the menu and click OK. Once again, type in a file name for the next subject. Flip back to the beginning of the printed ECG Lab Procedures manual and repeat subject setup, calibration, all 4 recording conditions, and then the saving steps for the next subject. You will repeat these steps for each subject recorded in your group. When your group is done recording all test subjects and each subject's ECG file has been saved to either a USB drive or the desktop, you may then select Quit from the BSL window to close out the application. Any files saved to the desktop should be now visible. If, however, files are missing, or you get to this point and realize you didn't save properly, there is a step in the saving segment of the instructions that details how to locate a backup file. In most cases, this step will work so long as the system hasn't been shut down. To access a backup file, select the File Explorer folder icon on the bottom of the screen. In the window that opens, select This PC on the left, then Local Disk C. From here, you'll select a series of folders starting with a Users folder, then Public folder, Public Documents, BSL Lessons, Data Files, and then a folder for each subject recorded. You can then copy these files to either a flash drive or to the desktop. For those with files saved to the desktop, you may now open an email account and send yourself your ECG recording file. To save time, you may also send a blast email to everyone in your group with all files attached. Once the email is sent from the recording laptop, each member of the group must then get a laptop from either the lab cart in the back or use their own laptop to begin the data analysis phase of the lab. Before proceeding to the data analysis segment of the lab, if there is another lab occurring after yours, please discard any used electrodes and leave all equipment attached and neatly arranged at the end of the lab table for the next group. If your class is the last to conduct the ECG lab for the day, follow the cleanup procedures listed at the end of the lab instructions. This entails discarding all used electrodes, shutting down the laptop and MP36 unit, wiping down the yoga mat before rolling it up and returning it to the designated cabinet in the lab, unplugging all cables, and carefully winding and neatly returning them to their labeled bin compartment in the BIOPAC satchel. And then returning the BIOPAC laptop with its power cable over it, as well as the BIOPAC unit and satchel to the cabinet in the front of the room where your professor can secure it. You can now proceed to the data analysis instructions in the printed ECG Lab Procedures manual provided. As previously mentioned, a PDF of these instructions is also available in Canvas under AMP2 Lab Resources if you do not finish analysis in lab and need to complete it at home. All of the laptops in the lab cart have the BSL software installed and may be used to complete the post-lab. It is recommended that you watch the ECG data analysis tutorial video on the MC Anatomy Physiology YouTube channel for step-by-step instructions on how to open your ECG file and complete the ECG post-lab in your must-know book. A link to the data analysis tutorial is available at the end of this video, as well as in Canvas.